Hello and welcome to G Fun Facts Online, your home for the most amazing facts from across the science world. Let's start with a question. Do you think of your brain as a computer? A piece of hardware that you get when you're born and that's it? For more than 100 years, that was the dominant view in neuroscience. The adult brain was seen as a fixed, finite entity. A masterpiece of engineering for sure, but a finished one. The central dogma was that we are born with all the neurons, all the brain cells we will ever have. This idea shaped everything, our entire understanding of learning, of memory, of aging, and the tragic permanence of brain injury. It was a powerful and frankly a somewhat pessimistic view of our own potential. But what if I told you that this foundational belief, a bedrock of neuroscience for a century, has been completely and utterly overturned? What if your brain is not a static piece of architecture, but a dynamic, thriving, and constantly renovating city? A city that can build new structures, forge new pathways, and even create new citizens, new neurons, throughout your entire life. Today, we are journeying into one of the most exciting and hopeful discoveries in modern science: adult neurogenesis. This is the story of the brain's remarkable and once unthinkable ability to generate new neurons from birth until old age. It's a paradigm shift that has opened up what many call an endless frontier in our quest to understand our capacity for lifelong learning, adaptation, and repair. So, get ready to meet the new you, or rather, the constantly renewing you, as we explore the incredible plasticity of the human brain. To appreciate how revolutionary this discovery is, we have to understand the dogma it replaced. The no new neurons doctrine held an iron grip on the scientific world for decades, and its roots go back to one of the fathers of modern neuroscience, Santiago Ramon y Cajal. He was a brilliant scientist who won the Nobel Prize in 1906. His neuron doctrine correctly identified that our nervous system is made of individual cells called neurons, not a continuous web. But he also stated, quite definitively, that in the adult brain, the nerve pathways are fixed, ended, and immutable. He believed that everything could die, but nothing could be regenerated. And when a figure of that stature speaks, the world listens. For the better part of a century, that was the end of the story. Then came the whispers of rebellion. In 1962, a scientist named Joseph Altman at MIT was conducting experiments on rats. Using a brand new technique to label newly divided cells, he saw something that should NT exist: evidence of brand new neurons being born in the adult rat brain, specifically in the hippocampus, a key area for memory. He published his findings in prestigious journals like Science, and the response from the scientific community was a resounding silence. His work was met with deep skepticism, largely ignored, and dismissed as a potential artifact of his technique. For 20 years, Altman's groundbreaking discovery languished in the footnotes of neuroscience. But the truth has a way of bubbling to the surface. In the 1980s, the evidence started to become undeniable. Shirley Bayer, building on Altman's work, confirmed that these new neurons were indeed being added to the adult rat brain. And then came a truly elegant piece of evidence from Fernando Nottebohm at Rockefeller University. He was studying canaries. He noticed that male canaries learn complex new songs every single year to attract mates. He wondered, could their brains be changing to accommodate this new learning? What he found was astonishing. The number of neurons in the part of the canary's brain responsible for song control dramatically increased during the mating season, precisely when they were learning new music. It was a direct, functional link. New learning was happening, and new neurons were appearing alongside it. This was hard to ignore. The final pieces of the puzzle fell into place in the 1990s. In 1992, scientists finally identified the source of these new neurons, neural stem cells, lurking within the adult brain. These are master cells, capable of dividing and transforming into new brain cells. This was the smoking gun. And then, In 1998 came the definitive proof in humans. Scientists studied post-mortem brain tissue from cancer patients who had received injections of a substance called BRDU, which gets incorporated into the DNA of newly dividing cells. When they examined the brains, they found the BRDU marker inside brand new, fully formed neurons in the hippocampus. The dogma was officially dead. Humans, just like rats and canaries, could create new brain cells throughout life. So if our brains are building new neurons, where are the construction zones? As we mentioned, this isn't happening everywhere. It's restricted to very specific, highly specialized neighborhoods called neurogenic niches. Think of them as the brain's exclusive, 5-star nurseries, providing the perfect environment for new life to begin. The first and most studied niche is the subgranular zone, or SGZ, nestled deep within the hippocampus. The hippocampus is that famous seahorse-shaped structure that acts as our memory-making headquarters. It helps us form new memories of events, facts, and places. The neurons born in the SGZ are mainly granule cells, and their job appears to be intimately tied to the hippocampus memory functions, especially in forming brand new memories and helping us distinguish between very similar experiences. The second construction zone is the subventricular zone, or SVZ, which lines the walls of the lateral ventricles, the large, fluid-filled chambers in the center of our brain. Neurons born here are destined for a great migration. They travel along a pathway called the rostral migratory stream all the way to the front of the brain, to the olfactory bulb. This is the brain's hub for your sense of smell. Once they arrive, they integrate into the circuitry and help you process and remember different smells. Imagine smelling a specific perfume that instantly transports you back to your grandmother's house. These new neurons are part of the machinery that makes that possible. And research is even beginning to suggest other potential niches, like in the amygdala, the brain's emotion center. These niches are more than just a location. They are complex ecosystems, rich with blood vessels delivering nutrients and oxygen, and surrounded by a host of support cells that release all the right signals to tell a stem cell to wake up, divide, and become a brand new neuron. The journey from a simple stem cell to a fully functional, integrated neuron is an epic saga of survival, migration, and connection. It's a multi-stage process that is both beautiful and brutal. It all starts with the division of a single neural stem cell. This gives rise to progenitor cells, which are like teenagers who have a better idea of what they want to be when they grow up. In this case, a neuron. These progenitors then mature into neuroblasts, which are basically baby neurons. But a baby neuron is useless on its own. It has to join the society of the brain. To do that, it must complete its journey. In the hippocampus, this is a relatively short move from the SGZ into the main layer of granule cells. But for the neurons born in the SVZ, it's that long-distance trek to the olfactory bulb. Once it arrives at its final destination, the neuroblast must differentiate. It sprouts an axon, the long cable for sending signals, and a beautiful, branching tree of dendrites for receiving them. It takes on the shape and chemical properties of its neighbors, trying to fit in. But the most critical test is synaptic integration. It has to successfully plug itself into the existing network. It needs to form thousands of connections, or synapses, receiving information from some neurons and passing messages along to others. Only then can it participate in the brain's conversation. Now, here is the harsh reality of this process. The vast majority of these newborn neurons don't make it. A significant portion of them are programmed to die within the first few weeks of their existence. It's a true survival of the fittest, or, more accurately, survival of the most useful scenario. Their survival depends almost entirely on whether they are needed. If they are stimulated, if they are recruited into a new learning task, if they make strong connections, they get to live. If they sit around idle, the brain's quality control systems will eliminate them. They are essentially on probation, and they have to prove their worth to earn a permanent place in the brain's circuitry. This probationary period is where things get really exciting for us, because it means we have a say in the matter. We can influence which of these new neurons survive and thrive. The rate of neurogenesis is incredibly plastic, shaped by our actions, our environment, and our state of mind. Let's look at the positive regulators, the things you can do to boost your brain power. The first, as we've hinted, is learning. But not just any learning. Effortful, challenging cognitive stimulation is key. When you push yourself to learn a new skill, like playing the guitar or speaking a new language, you are essentially sending out a help-wanted ad to these new neurons. You are telling your brain, hey, I'm building new circuits here, and I need all the hands I can get. This effortful learning process seems to rescue the young neurons from their programmed death sentence and helps them integrate. So it's a virtuous cycle. Learning promotes the survival of new neurons, and those new neurons then enhance your ability to learn even more. Number 2 on the list is one of the most powerful tools we have: physical exercise. Specifically, aerobic exercise like running, swimming, or even brisk walking. The evidence for this is overwhelming. Exercise increases blood flow to the brain, which is great, but it also triggers the release of a key molecule called brain-derived neurotrophic factor, or BDNF. You can think of BDNF as Miracle-Gro for your brain. It supports the health of existing neurons and powerfully stimulates the birth and survival of new ones. Another powerful booster is living in an enriched environment. For lab animals, this means bigger cages with toys, running wheels, and friends. For humans, it translates to a life filled with novelty, social engagement, and diverse experiences. It means breaking out of your routine, traveling, trying new foods, having stimulating conversations, and visiting museums. It's about providing your brain with a constant, rich stream of new information to process. And of course, there's diet. What you eat directly impacts your brain's nursery. Diets rich in flavonoids, found in things like blueberries, tea, and dark chocolate, and omega-3 fatty acids, which are plentiful in fatty fish like salmon, have been shown to promote neurogenesis. Conversely, some lifestyle factors are potent inhibitors. The undisputed public enemy number 1 of neurogenesis is chronic stress. When you're constantly stressed, your body pumps out stress hormones like cortisol. High levels of cortisol are toxic to the hippocampus. They suppress the creation of new neurons and kill off existing ones. This is thought to be one of the core biological reasons why chronic stress leads to memory problems and is so tightly linked to depression. As you might expect, the rate of neurogenesis also naturally declines as we get older. But again, the story is a hopeful one. It doesn't drop to zero, and an older brain can still be stimulated by all the positive factors we just discussed: exercise, learning, and enrichment, to keep the neuron factory running. So we have these new neurons, and we can influence their numbers, but what is their unique contribution? Why does the brain go to all this trouble? Their function is a hot area of research, but we have some compelling ideas. One of their most critical jobs appears to be pattern separation. Our hippocampus is amazing at storing memories, but it faces a challenge. How does it store memories of very similar events without getting them mixed up? For example, think about having lunch at the same cafe with the same friend every week. How do you remember the specific conversation you had this Tuesday, as opposed to last Tuesday? Pattern separation is the process that allows your brain to tag each memory as unique, focusing on the subtle differences to keep them distinct. Newborn neurons, because they are young and highly excitable, seem to be perfectly suited for this job. They are more plastic and can be molded to encode the unique details of a new experience, preventing a blurry overlap with old memories. This is also related to the famous spacing effect in learning. You've probably noticed that you learn better when you space out your study sessions over time, rather than cramming all at once. Research has shown that this spaced learning method not only enhances your memory, but also significantly boosts the survival of new neurons. It seems that spacing out the learning gives these new cells the time they need to mature and properly integrate into the memory circuit. Beyond memory, there's a strong link to mood regulation. The connection between low neurogenesis, stress, and depression is so strong that many scientists now believe that boosting neurogenesis is a key mechanism for how antidepressant treatments work. By creating new neurons, we might be increasing the brain's resilience and flexibility, making it better able to cope with stress and regulate mood. Finally, and most directly, a groundbreaking 2024 study provided the first direct evidence in humans that our ability to form new verbal memories, like remembering a conversation you just had, is critically dependent on the generation of new brain cells. It's not just a nice-to-have feature, it's essential for how we learn and remember in our daily lives. The discovery of adult neurogenesis is more than just a cool fact. It has fundamentally changed our view of the brain from a static machine to a living, dynamic, and perpetually renewing organ. It opens the door to incredible therapeutic possibilities. In the future, we may be able to develop treatments for neurodegenerative diseases like Alzheimer's or Parkinson's that don't just slow the disease but actively repair the brain by coaxing its own stem cells to replace what's been lost. We may have entirely new classes of drugs for depression and anxiety that work by targeting the brain's neurogenic niches. We could even develop therapies to significantly enhance recovery after a stroke or a traumatic brain injury. This research is a beacon of hope. But the most powerful part of this story is that you don't have to wait for a futuristic therapy. You can start applying this knowledge and nurturing your brain's endless frontier right now. The power to build a better, more resilient brain is, to a remarkable degree, in your hands. So let's recap the key takeaways. 1. Never stop learning. Be curious. Actively challenge your mind with new and complex tasks. The more effort you put in, the more you signal to your brain that it needs to keep those new neurons on the payroll. 2. Move your body. Make aerobic exercise a non-negotiable part of your routine. Think of it as fertilizing your brain's garden. 3. Embrace novelty and enrich your life. Step outside your comfort zone. Your brain thrives on new experiences, new people, and new environments. 4. Master your stress. Chronic stress is the arch nemesis of a healthy brain. Find healthy coping mechanisms that work for you, whether it's mindfulness, meditation, yoga, or simply taking a long walk in nature. And 5. Eat a brain-healthy diet. Fuel your mind with the nutrients it needs to thrive. The journey into the secrets of the brain is far from over, but the discovery of adult neurogenesis gives us all a profound sense of agency over our own minds. It's a powerful and optimistic reminder that we are not fixed in stone. We are constantly changing, and our potential for learning, growth, and renewal truly lasts a lifetime. Thank you for joining us on Fun Facts Online.